Hello, Stéphane.
Hello. So it seems that that's time now. Hello, everyone.
Hello, everyone.
So nice to attend today and to welcome you at this part 1 microbiome webinar from Biofortis, new efficacy and toxicity, endpoint in clinical studies. So the agenda for this half an hour, that's to briefly present you who we are, what is Biofortis, then to have the presentation of our colleague, Miss Dr. Françoise Le Vacon on exploring the microbiome, then to dedicate a time for questions. Please use on the toolbox the question tab you have to fill in, and we will recover these questions to submit to Françoise these points. So next slide. A short overview. We are very proud to be a part of the Mérieux legacy. So Biofortis, that's a company within the Institut Mérieux with our footsteps of Louis Pasteur microbiology, serving medicine and public health across the globe. That's a corporation with 13,000 of people, with Biomérieux, Mérieux NutriScience. We are in Mérieux NutriScience and Transgene, so 3 bio-industrial companies. What is Biofortis? We are a CRO, contract research organization, dedicated to clinical research, clinical services, and bioanalysis. Central lab services with a big focus on microbiome monitoring with our own next generation sequencing platform. That's very important also as a clinical microbiome companion to consider that we need and we have our own R&D unit. Research programs are developed by the way of actions from Miss Françoise Le Vacon to promote new technologies, new methodologies to serve our company tomorrow for clinical and central lab services. What we do, we attend the scientific services with clinical and microbiome services to promote and help companies for health food products like dietary supplements, functional ingredients, but also FSMP, medical devices, drugs, cosmetics, to prove and assess the safety, efficacy, tolerance, dose response, and bioavailability of these products, with one key point to submit to health competent authorities like EFSA, EMA, FDA, CFDA, or for instance, Health Canada. Biofortis in the world, that's three spots, 3 main locations in France. This is a headquarters with 2 private owned investigation centers, one dedicated central lab platform, 1 investigation center and also CRO part in the USA, Chicago, and subsidiaries in China. 120 collaborators, specialists, researchers covering these 3 main regions to serve innovation. Our clinical and analytical service under the cover of ICH GCPs, good laboratory practices, using ISO norm and a very high level of quality. Since 2002, that's more than 500 projects, 250 full service for clinical management in whatever the health field, metabolism, gastroenterology, weight management, oxidation, satiety, etc. Our job in the concept of one-stop shop or plug and play, that's to design the protocol, clinical protocol, until the data valorization, by the way of regulatory aspects, management of the studies, site management, bioanalysis, data science, and biostatistics. So I'm very proud to participate and introduce my colleague, Dr. Françoise Le Vacon. Françoise is Chief Scientific Officer and co-founder of Biofortis. This is she is an entrepreneurial woman for sure, and a specialist in cellular and molecular biology, cell immunology with a degree of PhD. Worked and collaborated with the Institut Pasteur, Johns Hopkins Hospitals, NIH, and very dedicated to biomarker discovery, oncology, microbiology over the last 20 years. Françoise is our microbiome research program leader for preclinical and clinical applications. Thanks, Françoise, to explain us what is the microbiome.
Thank you, Stefan, for the nice introduction. So the topic today is the microbiome, new efficacy and toxicity endpoint in clinical research. So after a short introduction, I divided my presentation into 3 parts. In the first one, I will try to speak about a healthy and a healthier microbiome, then how to design a clinical study, the volunteer side, and after the microbiome side. So, over the last few decades, we have observed an acceleration of knowledge about the microbiome, with an exponential increase of scientific communications, but also in the number of clinical studies, as we can see on the graph to the right. Here, only the studies recorded on clinicaltrials.gov, but the main. Thanks to advances in sequencing and computational biology, and specifically with preclinical studies, we're also making progress in the mechanisms involved. So recently, Berg reported in a review the consensual vision of experts gathered in the framework of the microbiome support project in Austria in the last year. The microbiota is a collection of microorganisms. Its composition is not limited to bacteria and contains archaea fungi. The microbiota includes all living members of the microbiome. Phages and viruses are the most controversial points of the definition, while the microbiome include their theater of activity, the constitutional elements of microbe, proteins, lipids, saccharides, and nucleic acids, the mobile genetic elements, the microbial metabolites, and all embedded in the environmental conditions of the habitat. So the human microbiome is elaborate and dynamic. Trillions of microbes live commensally on and within several tissues and biological fluids, including the digestive, urogenital, respiratory tract, the skin, mammary glands, etc. The placenta and brain microbiome are still controversial. Each site has its own microbial diversity. It's highly diversified, individualized, and initial colonization of the infant sets the stage for a relatively stable adult microbiome, the core microbiome. For bacteria, it could represent 20% of the bacterial genera. So the microbiome host interactions are complex, and we face a high degree of inter-individual variation. So now, what about and how can we define a healthy and a healthier microbiome? First of all, human health is not only no disease, but a complete well-being in 3 dimensions. The first dimension is physical and physiological, the genetic background, the diet, the medication. The second dimension is mental, the working stress or living conditions. The last is social, with education and revenues. Over the last few decades, we rediscover an ecosystem, a microbiome at the service of our health. 30% small molecules found in the human blood are derived from microbes. The gut microbiota clearly influences the health of its host. It provides crucial benefits in the immune system development, prevention of infections, nutrient acquisition, and perhaps even brain and nervous system functionality. It plays a crucial role in the development of pathologies such as obesity, autoimmune, and neurological diseases. In this context, we face a challenge to define a normal or a healthy microbiome. Rutledge says one person's healthy microbiome might not be healthy in another context. Example, if a person from the northern hemisphere moves to the southern hemisphere, they are healthy. In the same train, Aaron Segal said there could be multiple healthy configurations. And for Jason Lloyd Price, in a healthy person, the microorganisms coexist peacefully. Joël Doré and collaborators addressed this issue. One setup, the equilibrium of the microbiome is more or less modified by external events, diet, lifestyle, and particularly infections and drugs. Associations were described between major chronic diseases listed here and this biotic signature. Some pathologies can be linked to a disruption of the ecological balance and overall symbiosis. So a good definition of dysbiosis could be the disruption of microbes host symbiosis. So a healthy microbiome is well balanced and diversified. A healthier microbiome is a microbiome that you want to restore, reconfigure, an ecosystem more robust, rich, and resilient. So to demonstrate that we want to improve the microbiome, this exercise is easier in patients with infection or after antibio or chemotherapy. It's more difficult to demonstrate in general or healthy population. So this can be done by using a variety of assessments fairly widely recognized. Having a more diverse community, a higher microbial richness, hygiene count, increase the abundance of selected symbionts, such as lactobacilli or bifidobacteria species, Akkermansia mulcinifila or Fecalibacterium prosnitzi. Or also you can demonstrate the reduction of pathobionts or pathogens and the production of certain metabolites such as short-chain fatty acids. And now, how to design your clinical study? First, the volunteer side. Regulatory side, first, reminder that the regulatory status is defined by the intended use of the product. Food, drug, cosmetic, live biotherapeutic products. That means that one product can fit in several regulations. As example, an anti-dandruff shampoo is both a cosmetic and a drug. Second, claims vary widely across the globe, in Europe, in US, Brazil, or locally. Concerning food, many regulatory frameworks are intended for the general or healthy populations. So no many health claims related to microbiome. They are more common outside North America and Europe. As example, in Singapore, you can find the claim, probiotics help to maintain a desirable balance of beneficial bacteria in the digestive system. Regarding the LBPs, the FDA has established guidelines, but no LBP products have been approved as yet. 2 products are in phase 3. In Europe, the Pharmabiotic Research Institute prepared a project with the MMPs for microbiotic medicinal products, including LBPs, but also non-living products, phages, fecal material, or consortium-derived products. So if we focus on the last guidance of the European Food Safety Authority about the health claims related to gut and immune function, clinical outcomes can be accompanied or not by evidence of a beneficial physiological effect in combination with microbiological data. The guidance does not provide an exhaustive list of beneficial physiological effects, and the study's outcomes are not really defined. I chose two applications for which the microbiological study is sufficient. The 354 article, the claims on defense against pathogens in the lower urinary tract. So microbiological data could be used instead or in replace clinical outcomes. And in the 355, the claims on defense against vaginal pathogens can be achieved by increasing the proportion of lactobacilli and or decreasing the proportion of potentially pathogenic bacteria and or yeast in the vagina. We will note that these microbiomes are less complex than that of the gut. And there is only one article regarding infection in the gut area. So now, what are the key points of the clinical design? First of all, addressing a clear scientific question will define the methodology of the clinical trial. First step is to define and characterize the target population, the age, subjects, sex, life stage, their clinical course of the pathology, the physiopathology, and the homogeneity of the population, if you have some groups, the cohort size, of course, and relevant controls. The exclusion criteria are very important to define according to the study. Recent infection, gastrointestinal medication, alcohol consumption, smokers, taking supplements, drugs, loss or weight gain, diarrheal diseases in the last 3 months, etc. In microbiome research, many confounding factors need to be documented. First, diet and treatments, antibiotics, PPIs, antidepressants, anti-inflammatories, and the transit time also. You can also take into account some covariates, sleep cycle, menstrual cycle, fat-fasted cycle, metabolic status, hydration status, etc. A lot of data is needed, but the questionnaire must remain reasonable and easy to use. Of course, you have to choose the type of study protocol. To assess the recruitment or the intra or inter-individual variability, you can start by a pilot study with a few dozen persons. As the subject is his best control, as far as possible, the randomized crossover study is the most favorable. Otherwise, turn to a double-blind placebo-controlled trial. Concerning the product, the level and duration of exposure can be redefined during a pilot study, for example. More delicate is to define the time points. This should be discussed with your CRO as well. The number of visits, the sampling and its frequency, baseline versus end of treatment, intermediary checkpoints, the duration of study follow-up, the need to make sequential microbiome sampling. A lot of questions. So now, how to design your clinical study, the microbiome side. In my study, what do I want to demonstrate? Is my product improves or maintain, or is less toxic to the microbiome? Is it capable of restoring microbial diversity? Can I follow specific populations, phylum, specific strains? To answer, we will have several approaches. First, to analyze the structure of the microbiota by a study of the taxonomic composition by metabarcoding. We can analyze the microbial diversity, that is 3 components: richness, evenness, and disparity, or quantify some microbial population by quantitative metagenomics, qPCR or flow cytometry. And to assess the functions, we can study the hypothetical microbial functions with metagenomics, such as metabolic pathways or specific genes, resistance or virulence gene as example. But microbiota analyses are not sufficient. For a better understanding of the host microbiome interactions, it's fundamental to complete this analysis with other markers. After routine physicochemical parameters, such as stool consistency, frequency, water content, pH, etc., we can analyze complementary biomarkers, such as short-chain fatty acids, biliary acids, but also inflammatory markers, such as secretory IgA, fecal calprotectin, plasma neopterin, and also you can assess the permeability with the marker zonulin. And with other omics, such as metabolomics, for example, you can address how does the community respond to its environment. When analyzing different parameters, we are confronted with basic technical difficulties, but we have to anticipate, based on our strong experience, we have to consider the stability of the different parameters. Between -2 and 8, your stool sample is stable 7 days for calprotectin, but only 2 days for metagenomics. Our recommendation for the stool device is to use an anaerobic system. It's user-friendly and sufficient for metagenomics. And be careful, because devices with conservative are very expensive and not adapted for biochemical markers. Regarding the meta sequencing, we have to consider that the raw reads number can be completely different from the final usable reads number. We sometimes select that 5% of the raw dataset. Whatever the DNA extraction method, final sample will contain host DNA, so you lose reads, particularly in the skin flora. Whatever the sequencing platform you use, inside readers make sequencing error. Depending on the quality controls, you lose more or less reads. When you get clean reads, you have to assign them, and depending on the reference of the database and algorithm, at the end, will you have enough usable reads for your project? Different sequencing depths are possible, from very low to very high depth. The medium depth level with several million reads, which is named the advanced level in Biofortis, because according to the literature and to our expertise, it represents the best balance for taxonomic and functional microbiome studies. This level can be useful for secondary and exploratory endpoints in a clinical study. So, to analyze the omics, clinical and biological data and extract usable data from it, we need specific tools. And my colleague will tell you about it in the second part of the webinar. So now my take-home message is for clinical research. Microbiome studies need an appropriate experimental, methodological, and statistical design. It's necessary to associate the microbiome outcomes and different biological markers. For the design and conduct of your studies, it's necessary to get in touch with a microbiome expert partner. Finally, don't forget the part 2 of our webinar. So thank you for your attention. Stéphane, are you here?
I am here, but I was muted. I'm not now. Thanks, many thanks, Françoise, for your help first, and many thanks for this very interesting presentation. I hope this was very useful for our attendees for better understanding of the role of microbiome as an assessment parameter to include in the developments. We have some questions. First, please consider that you will have a replay of this webinar in a couple of days, so you could see again this presentation, and you understood that you will have part 2 of this microbiome webinar. We have some questions, several ones, so we will try to submit these ones to Françoise. First, Françoise, do you hear me?
Yes.
Okay. First question: is there an ISO standard or ICH guidelines outlining recommended protocols for microbiome testing? That's my question. No, that's a question.
Okay, thank you for this question. No. There is no ISO standards. Some labs follow the IHMS recommendations, others those of the HMP. But 2 founders of these guides, Joël Doré and Lita Proctor, are gathered together in an international initiative which mobilized the academies of science and medicine. One of their goals is to publish in a consensual white paper with the definition and recommendations for microbiome research. Biofortis is mandated to coordinate this program, so you can contact me if you wish more information.
Thank you. Second one, you mentioned genomic analysis, but what is the regulatory status of the human microbiome? Sorry, does microbiome analysis fall into the same ethical rules as human genetics?
No, there is no status. There is a legal gap. This, of course, this raises the issue of IP. So the microbiota is specific to each individual, but doesn't belong today to its host. So, for example, in the case of the development of probiotics or LBPs, an informed consent of the volunteer is sufficient and does not entitle him to fees. So maybe we will see in the future.
Okay, so perhaps 2 or 3 other questions. One, it seems that currently the complexity of microbiome data allows us to get out what we want depending on the statistics used. Do you have specific recommendation on this point?
Indeed, with various tools, we can deduce robust and exploitable results, but this is precisely the subject of our next webinar on microbiome data. So I will let my colleague to speak about that.
Okay, so we are
it's not my specific expertise, so
please call me back. Several questions. We have time. The given definition of abiosis question about the microbiota representative. Sorry, I have a little bit. Okay. The given definition of abiosis question about the microbiota representative of a control group, when a microbiota can be healthy is one condition and not anymore in another, what can we consider as control healthy microbiota? Is this question clear for you, Françoise?
It's a question I'm not sure to understand, but that means that it's difficult to define the controls.
Yeah, the healthy.
Yes, in the
what is the standard
trials.
microbiome?
Yeah, of course, the controls must be in the same area, in the same country, in the same area. As I mentioned during the webinar, people from Sweden, for example, in another context, the microbiome can be in disruption and not really is healthy. The subject is healthy, but the microbiome is in this biosis. So it's not easy to define the good control groups. First, we have to discuss about the endpoints you want to demonstrate. And of course, the healthy microbiome is not so easy to define because there is no score, no really standardization of the threshold and health score. There is some publications that try to postulate with some health score with a set of thousands of microbial species. If you have these microbial species, you can say that you are healthy or with a level of diversity, but there is no consensus today. So depending of really depending of the clinical trial.
Okay, so I will.
It's a complex, yes, it's a complex question. Yes.
Yeah, so I have got another complex question, Françoise. Thanks to Sylvie, Lamia, Kadi, Sylvanie, Irini, Wilfried, Mavelin, also to ask questions. Please be confident we will reply on your question, but second time by writing, okay? But this is a global question, Françoise. One of Sylvie. Did we already define what is the standard microbiome?
No. That means standard. That means more communities to use in the analysis. Maybe I don't know if she means yes. We have some more communities as controls in the workflow, in the analytical workflow.
Like
yes,
yes,
it's the first step to the standardization.
Yeah, because the standardization process is ongoing, everybody.
Yes, we have more and more the workflow are well. We know the BS doing all the analytical workflow. So maybe you have to have really robust procedures. But we know also that depending on the lab, the microbiome can be different. It's a reality.
Thank you, Françoise. May I have to ask you two other questions, Françoise?
Okay.
Do you agree?
As you want.
Okay. A technical one. Is your opinion that fecal samples older than 12 months at -80 should not be analyzed at all if their stability is questionable? A question about -80 samples and their
versus room temperature, or
no, after 12 months, what should we say about the stability of stools, for instance? And
yes, for collection, for stool samples, you need to put them 80 for collection. After, depending on your project, you can use some specific device usable at room temperature, but you know, it's for specific analysis, and depending what you want to do with your sample. But for a collection of 2 samples, the best today is -80.
Okay, and the last one, please, all of the attendees, we will share the answers of our experts in the second time. Would you consider, Françoise, the microbiome as a cause or a consequence of a pathology? From Sylvanie.
It's
No,
really, I can answer because we know more and more about the mechanisms, and we know that we can transfer some disease such as obesity with a transfer of microbiome, but in a lot of cases we don't know if it's a cause or a consequence. So we have some evidence in some pathology. We know that some bacteria really can trigger a disease, in infectious diseases or in some colon cancer, for example, but in a lot of cases we don't know. We know that we have some associations, a lot of association between the microbiome and the disease, but we need more and more in vitro and in vivo studies to demonstrate in different cases. But we have some evidence in some pathologies.
Okay, so thanks a lot, Françoise, for this exchange, for your support, for microbiome support in Biofortis. Thanks a lot. Thanks a lot, everybody, because we had a lot of questions and a very interactive session. We are very happy of that. Take care of you, and please call back. Okay. So have a nice evening for European and a good day for US. Bye-bye.
Bye-bye.